Hello, everyone. My name is Kara Laney, and I'm the study director for the National Academies of Science, Engineering, and Medicine, and the study director for this particular study on exploring the linkages between soil health and human health. I am substituting as host today for our committee chair, Diana Wall, who was called away at the last minute. I will be moderating our Q&A today with our three speakers. But first, I'd like to just take a few minutes to tell you about the study process and give you an overview of today's format. We will be hearing from three speakers today, and we will adjourn at 2 p.m. or when we are done with our Q&A, whichever comes first. I want to start with a note that this is an open on-the-record session. Interested individuals and members of the press have been invited to join as observers, but this is not a news conference and we will not be entertaining questions from the audience. Reporters who would like to talk to members of the committee are kindly invited to touch base with me. And anyone who would like to submit a written document or comment to the committee is also welcome to do so on the study's website by clicking the Provide Feedback button on the right side of the website. You can also subscribe for updates about this study by signing up for the Board on Agriculture and Natural Resources listserv, which is also on the right side of the page. I would like to remind everyone that this is an information gathering session and that the committee is in the process of assembling its materials that it will examine and discuss in the course of making its findings, recommendations, and conclusions. I ask everyone to be mindful that the committee has made no conclusions, and it would be a mistake for anyone to leave this meeting thinking otherwise. Comments made by individuals, including members of the committee, should not be interpreted as positions of the committee or of the academies. In addition, committee members typically ask probing questions in these information gathering sessions that may not be indicative of their personal views. The committee will deliberate thoroughly before writing its draft report, and once the draft report has been written, it must go through rigorous review by experts who are anonymous to the committee. The committee must then respond to those comments in a way that satisfies the National Academies Report Review Committee and the NAS president before it is considered an official Academy's report. Our committee on exploring linkages between soil health and human health is comprised of 15 volunteer experts. Their names and university or business affiliations are here on the screen. You can find a short biography of each of them on the study's website. Now I'm just going to walk through a few bits about the National Academy study process for those who might not be familiar with it, and tell you a little bit about the National Academies more generally. So the National Academy of Sciences was established in a bill passed by Congress and signed into law by President Lincoln in 1863. Later, the Academy was granted the authority to create the National Academy of Engineering and the National Academy of Medicine, which was previously known as the Institute of Medicine. So today we're the National Academies of Sciences, Engineering, and Medicine, and the organization consists of the three honorific bodies as well as scientists across the country, inside and outside of the academies, who provide expert advice on the nation on issues involving science, engineering, and medicine. The mission detailed in the congressional charter is to report, examine, experiment, and report upon any subject of science or art whenever called upon by the government. We fulfill this mission through many convening activities, including consensus studies like the one we're discussing today. And just to share a little bit more about consensus studies as opposed to our other convening activities like workshops or forums, consensus studies are conducted by committees convened specifically for a unique statement of task. The experts who serve on these committees do so as individuals, not as representatives of any organization or institution to which they may belong, and they serve as volunteers. Consensus study committees review the state of the scientific literature to write a report that responds to the problem identified in the statement of task. And that report will hopefully be used to shape sound policies, inform public opinion, and advance the pursuit of the scientific field in question. Each consensus study goes through a number of stages. First, we make a statement of tasks, which we work out with the study sponsor. In this case, that's the USDA's National Institute of Food and Agriculture. Then we solicit nominations for individuals with the necessary expertise to respond to the statement of task. Nominees are appointed by the president of the National Academy of Sciences, Dr. Marcia McNutt, and that membership on the committee is provisional until the committee has completed a discussion of conflict of interest, disclosure, and composition at its first meeting. After being appointed, the committee moves into its information gathering phase, which is where we are right now. We invite in speakers to present to the committee either in person or via Zoom. Members of the public may also present information for the committee to consider. And as it gathers information, the committee is also beginning to deliberate on how best to address its statement of task. They will begin drafting the report, and when the draft report is complete, they will submit it to the report review committee, as I said before. When the report is final and cleared by the report review committee, it is delivered to the sponsors and made publicly available. I just want to say a few more words about the committee process. When we meet in person or in Zoom with members of the public, with speakers, with sponsors, the committee does so in open session. And anyone is welcome to attend these meetings. The committee meets in closed session in which only the committee members and staff from the National Academies are present. We do this so that the committee has the opportunity to deliberate and discuss the statement of task and change their minds as they may need to in a closed deliberative setting. This is the general timeline for the soil health and human health study. We're nearly done with the information gathering and meeting phase of this study. Then we'll be moving into finalizing the draft report and submitting it to review. We do have one more webinar planned for October 23rd, and we'll be posting information about that soon. And I anticipate that the report will be ready for release in the spring of 2024. Here is the study statement of task. I'll just pause for a moment. And next slide, Eric. So, as I said, the full statement of task can be found on the study's website, and the sponsor for this particular study is USDA's National Institute of Food and Agriculture. This study came to us from USDA through our congressional request in earlier appropriations legislation. Lastly, this is a screenshot of the study's website. So on the left, you can see where you can find the full statement of tasks, the committee members and bios, and information about our past webinars and when it's ready, the upcoming webinar. On the right, there's a link to provide feedback or comments on the study and a place to subscribe for updates about the study. If you need to contact the National Academy staff, my address as well as the program assistant's address are here on the screen. And with that, it's my pleasure to introduce our first speaker today. Virginia Moore is an assistant professor at the School of Integrative Plant Science at Cornell University. The research program focuses on plant breeding for sustainable cropping systems, encompassing breeding for organic systems, for intercropping and polyculture systems, for pest resistance, and for ecosystem services. She works in a large range of species, including cover crops, perennial forages, bioengineering crops, and hemp. She was previously a postdoctoral researcher and project manager for the legume cover crop breeding network at USDA's Sustainable Agricultural Systems Laboratory. Welcome, Virginia.
Thank you so much. And thank you to the committee for the work you're doing on this important topic and the opportunity to speak today. I'll get my screen shared. All right, can you see my slides? Great. So as Kara described, my lab at Cornell is focused on using plant breeding to strategically enhance cropping system diversity, among other sort of components of sustainable cropping systems. And today I'll provide a general overview of this topic area, breeding for complex systems, and some specific examples from the work currently happening in my lab. But first, to provide some context and about why we're focusing on complex and diverse cropping systems. So we've seen decades-long declines in crop diversity across the U.S. This figure represents the change over time in the proportion of land devoted to production of major crops in the U.S. over over nearly 2 centuries, and our acreage is now dominated by a few major crops such as corn and soybeans, with many others experiencing precipitous declines in acreage. These trends are present not only at the national scale, but also regionally. So this figure shows a measure of spatial concentration of crop production in the US. And that spatial concentration has increased fairly dramatically, especially over the last 30 years or so. So not only are we producing fewer acres of many of the more minor crops, but their acreage is also highly concentrated in smaller regions, making each crop more vulnerable to conditions within smaller areas. And farmers are generally also planting fewer crops in their rotations. Diversified crop rotations have historically been a tool that farmers have used to buffer against risks of crop failure, and also as a tool to manage pests and fertility and other aspects of their agroecosystem. So over time, we've seen that farmers are altering their risk management strategies, replacing those tools of crop diversity with agrochemical inputs such as fertilizer. This is fertilizer application rates globally, and then also with various pesticides. So this is global pesticide application just since 1990. And these shifts are bound up in technological and agricultural policy changes. And those changes have been very successful at increasing crop yields. But we know now that this change has also come with major costs for environmental and human health, which I'm sure this group is very familiar with. And plant breeding has been both a contributor and has also been shaped by these trends towards simplification and input dependence. Investments in plant breeding research and development has disproportionately focused on a few crops and production regions. And as plant breeders, our work tends to be conducted in these more simplified and monoculture cropping systems. But this focus on simplified systems isn't inherent to the technology of plant breeding. And I see opportunities increasingly for plant breeding to be used as a tool to develop cropping systems of the future that are both productive and diverse. So that's sort of the goal of my lab group and of many of our collaborators. And there's ample, and I would say growing evidence in the literature for ecosystem service benefits related to diversification. This is a figure from a recent meta-analysis that showed that in most cases that were examined in the analysis, that increasing diversification in cropping systems, kind of broadly defined, were associated with ecosystem service benefits and did not reduce crop yield. And I would say that plant breeding could be used to further develop and support adoption of these complex and diversified systems. So there's a few ways as breeders that we can do this. First, we can increase productivity within these diversified cropping systems. For example, there's compelling body of literature demonstrating that that crop varieties bred within organic systems tend to be better adapted to organic systems than conventionally bred varieties. And a common criticism of organic agriculture is that it's lower yielding compared to conventional agriculture and can't sustain current global production. But I would say that this challenge could be at least in part mitigated if organic producers had access to better adapted varieties for their systems and were therefore able to produce higher yields within those systems. We can also work to reduce barriers to adoption of diversified cropping systems. We can, I'll discuss shortly some examples of how we can do that, but basically enhancing traits that make it easier for farmers to use these systems. And we can also explicitly focus on enhancing ecosystem services, for example, promoting crops' ability to improve nutrient cycling or to sequester carbon. So in sort of practical terms, how might a breeding program address these breeding goals related to complex systems? And much of my lab's work is focused specifically on increasing crop diversity within rotations in various ways. So I'll focus on some examples of breeding within that context. So the first way that we can think about this is increasing temporal diversity. Farmers can increase diversity over time in their systems, growing multiple crops within a growing season or over multi-year rotations. And as breeders, we can think about breeding crops for new regions to increase rotation options for farmers. And this is, I would say, a relatively straightforward, low-hanging fruit pursuit that breeders can use to increase diversity. We already have the tools and species, and it's a matter of applying breeding in new environments and focusing on traits that are relevant for different regions than currently are a focus. So an example from my breeding program is our work on dry beans. So dry beans were previously grown across New York State on a larger scale, but production has over time shifted to other parts of the country, and there's relatively limited production in our state today. But we've seen that dry bean consumption is increasing in the US in recent years, and there's so interest in among consumers in supporting local farmers. And so New York farmers are interested in adopting this crop again and expanding production in the state to take advantage of this consumer interest. So along with collaborators at the University of Wisconsin, University of Vermont, University of Maine, we've been initiating work in dry bean systems for organic producers broadly. So there's some management components for this project, but we also have a breeding focus and a general focus of enhancing organic dry bean production in the Northeast and Upper Midwest. Our team started conducting variety trials this year in 2023, and we'll start breeding dry beans in the coming year. And in addition to breeding for this region and for organic production systems generally, we're also interested in examining genotype by management interactions across tilled and no-till production systems. So adding kind of a layer of rotational complexity there as well. In addition to breeding cash crops for adaptation to new regions, plant breeders can also increase temporal complexity by breeding cover crops or other crops aimed at ecosystem service goals. My lab has a major focus on cover crop breeding. Cover crops have numerous sort of environmental and management benefits on farms, and especially make a contribution to cropping system complexity by replacing time periods where we might otherwise have a fallow period, and when soils would otherwise be vulnerable to erosion and nutrient loss and other challenges that reduce soil health and quality overall. And we know that cover crop adoption has been increasing relatively rapidly in recent years, especially as more incentive programs promote their use. But unlike cash crops, there's been very little breeding effort focused specifically on cover crops, and farmers have few cover crop variety options to choose from currently, and relatively little information about the best variety choices among those that are available. So to address some of these challenges with cover crops, I currently lead the Cover Crop Breeding Network, which is a group of plant breeders, agronomists, and farmers who are developing and testing cover crop varieties across multiple different species. And we've we have breeding nurseries on research stations and on farms. So we run a participatory breeding network as well, and run a 15-site trialing network that spans the US. And given that cover crops have received this limited investment compared to major cash crops, in addition to developing new varieties, this network has been really powerful, also in generating kind of basic information about genotype by environment interaction and the zones of adaptation for cover crop species. And our breeding goals vary somewhat depending on the species, but generally fall into these two broad categories. We work on goals that are specifically related to enhancing ecosystem services provided by cover crops. So for example, we work on developing, across species, we work on developing cover crops that provide rapid cover in the fall. So we're mostly working on fall planted cover crops. So we're trying to increase the benefits protecting soil over the winter, so that we can maximize the ecosystem service benefits that the cover crops are providing. We also are working on breeding cereal rye for allelopathy, so the rye can provide improved weed suppression benefits. We also work on goals that are related more generally to reducing the barriers to cover crop adoption. So farmers consistently cite narrow planting windows and challenges with timing on the other end. So when they're terminating the cover crop and trying to get their next cash crop planted. So this narrow window of opportunity to use a cover crop is a big barrier preventing people from using them. And so we breed for things like earlier flowering and tolerance to late planting under colder conditions to reduce that barrier for cover crop adoption. And in addition to the temporal complexity that we can enhance through plant breeding, we also in my lab work on some different types of spatial complexity. So breeding cover crops for or breeding crops for variety mixtures and intercropping systems, and we also look at systems that involve kind of both temporal and spatial complexity systems where we have things like relay intercropping or perennial ground cover systems where part of the growing season we have monoculture and part of the season we have multi-species mixtures intercropping. And in general, when we're thinking about intercropping and breeding for these systems, we're trying to enhance cooperation among partner species and reduce competitive interactions. We may also be focused on characteristics related to the compatibility with different management requirements across species, such as the timing of planting or harvest or other management activities across species. In this first case, when we're thinking about competition, plant breeding has a role to play in reducing competition among species generally, with the idea that if we breed crops to fill different ecological niches in both time and space, which is in this figure represented along the x-axis, then competition between the two species represented by the overlapping area between the curves, that competition can be reduced, resulting in this overall increase in system function generally, which could be yield or quality or various ecosystem service characteristics represented along the y-axis. So we can see a decrease in that overlap as we do conduct selection as breeders and improve overall outcomes for the cropping system. One interesting relay intercropping system that my lab works on is cover crop interseeding between rows of cash crops. So this is a strategy that farmers use to overcome narrow windows available for cover crop planting, as I mentioned earlier. And the idea here is that the cover crop can be planted earlier in the growing season while the cash crop is still growing, and then have an advantage to establish before winter sets in, and there's a risk of cover crop death or winter kill. So in this system, ideally we have this establishment advantage, but in reality, even if cover crops are planted in an optimal window, survival is fairly inconsistent under the shade of the cash crop canopy. So in the context of my lab, we're working on intercropping with corn, and we're doing some breeding work with red clover. So we know that this is a pretty stressful environment for the cover crop, that it has to compete with the cash crop for light, moisture, and nutrients, and it's under this much taller canopy, and has challenges surviving until the cash crop comes off. So we've been breeding for improved survival under the canopy and looking specifically at shade tolerance as a mechanism to improve survival. And this is an instance when we're focused on sort of an end outcome related to corn productivity, and the ecosystem service benefits of the red clover are important, but ultimately secondary to the production system. But the goals in different intercropping breeding systems will vary depending on the partner species we're talking about. So when the intercropping system includes multiple cash crops, we might have a more equal emphasis on the yields of both species. And yeah, I guess I would leave the committee with this idea of the broader context that we're working in. There's a lot of complexity working with diverse cropping systems, complexity biologically, statistically, and logistically working in these systems. But I would say that we largely have the tools and conceptual frameworks we need to design effective breeding programs to deal with these challenges. But I think the larger obstacles are related to system drivers beyond plant breeding. There's for example, policy incentives that are still pushing farmers to use simplified cropping systems, and that limits system adoption and use of the varieties that we might produce for these systems. And likewise, we know that new crops and varieties have to be supported by markets for the resulting products, and that there's also a need for infrastructure and management systems that otherwise sort of support these diversified cropping systems. For a lot of the crops that we're working on, and that our collaborators are working on, the markets and infrastructure components are simultaneously with the breeding work we're doing. We're also working with stakeholders to imagine and develop the infrastructure components that are needed to make these systems work effectively. And I think, with these kind of complex, not straightforward processes, we really need interdisciplinary collaboration and participatory approaches involving farmers and stakeholders along the whole supply chain. In all of the projects that we work on, we're using some kind of stakeholder engagement, surveys, interviews, including stakeholders as formal collaborators on our projects. And so it's kind of the nature of the beast when you're working in these systems. And I think I'll leave it there and acknowledge my collaborators on our cover crop and bean projects and members of my lab who've pushed this work forward. Thank you.
Thank you, Virginia. We have time for a few questions for Virginia now, if anyone from the committee would like to jump in. Sarah?
Yeah, I can start. Sarah Collier at the University of Washington. Thanks for the presentation. That was fantastic. And I wondered, you mentioned at the end there about the need for interdisciplinary collaboration in this. And I wondered if you could just say a little bit more about what your experience has been to date or what you're looking at being needed in the future.
Sure, thank you. That's a great question. So I guess part of it, so my training and background is pretty interdisciplinary. I did work in plant breeding, but also in agroecology and ag economics. And so that's kind of working on these kind of complex problems that need cross-disciplinary work is kind of my comfort zone. And I have also brought in collaborators that have a more social science backgrounds that can lead the sort of stakeholder engagement work as well. And yeah, I've found it really fruitful to have, especially as we're working on new crops and new systems, to have sort of explicit stakeholder engagement components to the breeding projects that we're working on. We just finished up a survey and focus group process working to develop a new breeding project focused on organic hemp. And I found that process super fruitful, and I think will mean that the breeding work that we're doing later is more impactful. But yeah, I guess I would say, like, a lot of the systems that we're working in, especially figuring out what the end use product is so that we can kind of work back to understand what the quality and like other breeding goals should be from an end use processor end is really important. And making sure that we have, especially when we're thinking about intercropped systems, having equipment that is appropriate for planting and management and harvest of the crops is a big barrier. And I think there's some good work happening, especially at the Land Institute, where one of the next speakers is based. I think there's good work happening on that front. But it's, yeah, definitely an important part of the picture.
Mike?
Yeah, thanks, Virginia, for a very nice presentation and overview of your work. You kind of answered my question a little bit at your last statement, but I kind of have two questions. One, I'm curious, when you talk about the working more multidisciplinary, I mean, you've got these diverse cropping systems that you're working on. Have you been working with soil scientists in any way, and can you speak at all to how these more diversified systems might be impacting soil health? And on the other end, you kind of alluded to it, I'm curious about the various quality measures in the cash crops coming out at the other end of the system.
Yeah, that's a great question. So we haven't had engaging sort of any below ground characteristics has been a pretty new for our especially in the cover crop breeding side of things. We now are working with some root biologists to start looking at those traits, but have not. I would say, like, we've been hesitant given like the lack of sort of long-term data on using specific, like a single year plot, these annual species that were not growing in the same location multi-year, that we've been kind of limited in what we can say about their impacts on the soil. We know a lot about sort of like from an agronomic and soil science side, like what the impacts of these different management practices might be, but less about effects of individual genotypes, I would say. I think I see places for engagement with soil science in terms of helping us identify the characteristics that we should be breeding for, but yeah, I would say that's like we're at the early stages of incorporating that more explicitly. Yeah, and then can you repeat the second part of your question?
I was just curious about in these diversified systems, how that might impact the quality of the cash crops on the, you're harvesting onto these systems.
Yeah, that's, I guess, another place where we haven't done a lot of explicit work in like multi-year rotations and impacts of individual genotypes on a subsequent year cash crop. But yeah, I think it's a potentially fruitful avenue to explore, and we have I think there's some useful models of specifically focused on intercropping systems that I think could be applied in other contexts as well, where we look not only at sort of the effect of the individual genotype on yield or whatever other characteristic we care about, but like looking at effect on its neighbor or effect on a subsequent crop. Yeah, that we can evaluate and make breeding decisions based not only on the characteristics of the individual plant that we're selecting, but on on partners or on subsequent crops. I think it's for sure a useful thing to do.
Thank you.
Yeah.
All right, one more question from Rebecca, and then we'll go to our next speaker.
Great, thank you. Thank you so much, Jenny. That was really terrific. I've long thought of the diversification issues in relation to risk, which you bring up, and I'm wondering, the pathways between the kinds of risk that are under consideration and these health issues on the one hand, just because that's our topic at hand for the panel. Is it price risk and stress risk for farmers? Price risk leading to perhaps dietary impacts if food production is imperiled by all this climate chaos. But what are farmers, producers' attitude towards risk and the mitigation that the diversification offers? Do you feel people are welcoming this as a risk mitigation sort of line of work, or is it just more hassle than, like, do farmers see more hassle risk or more, how do your stakeholders sort of regard those risk issues, and do you feel like you're able to make a compelling case around risk management?
Yeah, I mean, I think it depends a lot on which farmers you're talking to, and yeah, there's definitely a hassle factor with these systems are knowledge intensive, and so executing them effectively requires a lot of trial and error and sort of understanding of the biology of the system that you're working in, and so it's not something that everyone is looking to pursue at this point. But I think that that a big part of it too is that, like, things like crop insurance are providing other tools to deal with the risk of a crop failure in an individual crop in a given year, right? And those tools are only available for a subset of the crops that farmers are planting, and don't necessarily account for sort of the higher value of organic products or other high value markets that farmers might be pursuing. And so I think that there are ways that policy could make sort of these complex systems a little more appealing, if there's kind of if risk management tools were also promoting diversity, if that's helpful.
Thank you so much, Virginia. Our next speaker is Jean-Michel. He is a professor at the Department of Bacteriology and the Department of Plant and Agroecosystem Sciences at the University of Wisconsin-Madison. His primary research interest is understanding the molecular mechanisms allowing efficient symbiotic associations between plants and microbes, and applying this knowledge to maximize the benefits of such associations in agriculture. Welcome, Jean-Michel.
Thank you. Thank you for this invitation and the work done by the committee. It's really a really important topic. So I'm going to talk today about efforts to try to engineer plant microbe symbiosis and for enhanced plant, soil, and human health. So as you know, we live in a microbial world. Whether we are humans, animals, or plants, we all interact with microbes. Plants interact with microbes at the root level, at the shoot level. Some of these microbes are on the surface of the plant. Some of them are inside of the plant. We call them endophytes. For my lab, I particularly focus on associations between plants and bacteria and fungi at the root level, and in particular on associations that we call mutualistic, that are beneficial for both the plant and the microbe. Typically, the kind of deal between the plant and the microbe in these associations is that the plant provides what plants are good at, carbon fixation. So plants usually provide a carbon source to the microbe. And in return, bacteria, fungi provide a wide range of benefits. Mycorrhizal associations and some bacteria are well known to help for water acquisition. Microbes provide nutrients necessary for plant growth, nitrogen, phosphorus, potassium. Microbes also help the plant for biotic and abiotic stress tolerance. So today I want to talk about one particular benefit. In my lab, we mostly work on nitrogen and water, but today I decided to talk about the nitrogen problem because there is actually a wide sort of diverse approaches to try to solve what I call the nitrogen problem in agriculture. So these benefits provided by microbes can be direct, but I also wanted to mention, given the scope of the committee, that in many cases these benefits to plants and humans are also indirect through the soil. The perfect example for that are some of mycorrhizal associations that modify the soil's texture, soil aggregation, that in turn help the plant for nutrient and water acquisition. But bacteria also modify the soil. So some of these benefits are direct, some are more indirect. Today, I'm going to talk about engineering some more direct effects. And in particular, I want to, as I said, focus on the nitrogen problem. I would be happy to answer questions about other benefits of microbes, but today I'm going to focus on nitrogen and in particular what we call nitrogen fixation. So to introduce the topic of nitrogen fixation, I come back to the nitrogen cycle, just as a reminder that most plants get their nitrogen from the soil, mostly in the form of nitrate and ammonium. What we call nitrogen fixation is the conversion of atmospheric nitrogen, where nitrogen is highly abundant, into ammonium, and can be done in different ways. You have industrial fixation, also called the Haber-Bosch process, invented at the beginning of the 20th century, that provides basically a lot of the synthetic fertilizers we use today, especially in high-intensity cropping systems. And these days, our agriculture is highly dependent on these nitrogen synthetic fertilizers. You also have atmospheric fixation happening. It's more minor every time you have lightnings. And today I'm going to talk a lot particularly about biological nitrogen fixation. So biological fixation is performed by bacteria and archaea. And you may have heard that some of our crops, like legumes, are particularly good at associating with some of these nitrogen-fixing bacteria. And that's why Virginia was talking about legumes. That's why we have legumes in all our cropping systems around the world, because they can associate with these nitrogen-fixing bacteria. Legumes require very little to no nitrogen inputs, and they can even bring nitrogen to the system. Unfortunately, outside of legumes, our crops, and in particular cereal crops, really do not associate well with nitrogen-fixing bacteria, and they require a lot of nitrogen to achieve good yields. Hence, that's what we call the nitrogen problem in agriculture. So to put numbers under the different types of nitrogen fixation that I was mentioning, well, atmospheric fixation is, as I said, relatively minor. Biological nitrogen fixation on land, so excluding the oceans, estimated between 90 to 140 million tons of nitrogen fixed per year, again, from the atmosphere to the soil. And if you're looking at that Haber-Bosch process, industrial fixation that allows us to make synthetic fertilizers, now it even exceeds biological nitrogen fixation on land. According to the latest number I was able to find, you see 170 million tons per year. So right away, you can see that obviously that new system is that new fixation, I would say, is obviously going to cause a disruption of the nitrogen cycle. But first, I need to say that the Haber-Bosch process and synthetic fertilizers are a good thing. In fact, I found that website that was trying to quantify how impactful different innovations of science have been for humanity, and they call that life-saving innovations. And synthetic fertilizers are the number one, are actually considered one of the most life-saving innovation. And for sure, because literally we can feed half of the world population with synthetic fertilizers. So here that graph here on the right shows you in blue the increase of the world population from the beginning of the 20th century to now. And in gray, the graph, that line represents the proportion of the population which is fed, sustained through synthetic fertilizers. And you can see that about today, we literally can feed half of the world with synthetic fertilizers. So obviously, when you think about human health, synthetic fertilizers are a good thing. We are feeding half of the world with fertilizers. At the same time, I'm sure that you have heard about the issues associated with fertilizers, synthetic fertilizers, and I wanted to remind you some of them to show you really the why I'm doing my research on biological fixation and why that impacts human health. Synthetic fertilizers are made out of natural gas. They are made out of a fossil fuel, natural gas, and they require a lot of fossil fuel. So we use natural gas to produce these synthetic fertilizers that then we use for food, feed, fiber, and fuel production. So when you think about what I just said before, that we are feeding half of the world with synthetic fertilizers, what that really means is that we are feeding half of the world with natural gas, with a non-renewable resource. So obviously, when you think long-term, it's not sustainable. We are feeding the world on natural gas, half of the world on natural gas. When you think that we are making these days biofuel using crops that we grow using synthetic fertilizers, then it's almost ironic, or at least I would say inefficient. So besides these long-term issues, there are also some more short-term issues that I wanted to remind you about. One is developing countries. The price of fertilizers basically is heavily dependent on the price of natural gas. And the cost of these fertilizers, in particular for poor farmers in developing countries, is an issue. And the fluctuations of the price of fertilizers is even more a problem. Here on that right graph, I show you basically the cost of fertilizers, of synthetic fertilizers over the last 3 years, where at the beginning of 2021, you see it was about $400 per ton. And then with the war in Ukraine, the price almost basically doubled, more than doubled. It stayed very high during 2022. And fortunately, these days it's coming down. But these fluctuations, and especially when fertilizers are so expensive, it's a significant issue for growers. The second issue in developing countries is the logistics, just the logistics of bringing fertilizers to these smallholders. I just wanted to mention that book from Roger Thoreau, The Last Hunger Season, that really shows very well these two issues for smallholders in Kenya. So that's one issue, price of fertilizers, access to fertilizer. In the US, the problem is more, the short-term problem, I would say, is more environmental issues. I'm sure that you have heard about nitrate leaching and eutrophication of streams and rivers, degradation of coastal zones and the Gulf of Mexico, all these direct consequences of the intensive use of fertilizers. And I don't know if you know, but fertilizers also contribute a lot, both because of their production, but also their degradation to global warming. And so for all these reasons, and actually, in fact, for quite a long time, the community, the scientific community realized that we should try to rely a bit more on biological nitrogen fixation. And in particular for crops like cereals that don't associate well with these nitrogen-fixing bacteria. And so that's what I want to talk about today, is what are some of these approaches that can impact not only plant health, soil health, and human health. So nitrogen fixation is performed again only by bacteria and archaea that possess an enzyme or an enzyme complex that we call the nitrogenase. And I don't want to go too much into the mechanisms of the nitrogenase, but I want to show you that figure for two reasons, to remind you two limitations of biological nitrogen fixation. One of them is ATP, or energy. In order to reduce one molecule of dinitrogen into ammonium, you need at least 16 molecules of ATP. It's a very energetic, expensive process. And so for any bacterium able to fix nitrogen, they will need access to a lot of energy. And in particular, when they associate with plants, that energy needs to come from the plant, usually in the form of a carbon source. The second issue of that these bacteria need to solve is the problem of oxygen. That nitrogenase complex has these iron cofactors that can be oxidized really quickly, and oxygen strongly inhibits the nitrogenase complex. And so in any efficient system where you have biological nitrogen fixation, there needs to be a system either coming from the bacteria or the plant to protect the nitrogenase and provide what we call a microaerobic or a low oxygen environment for these bacteria. And again, that can be done either by the bacteria or the plant, depending on the system you're considering. So what are the approaches these days that people are thinking about to try to have more biological nitrogen fixation in our cropping systems, and in particular when we grow cereals? Well, it has to do with plants and microbes. And so some approaches are more oriented on the plant side and some more on the microbe side, and some are doing both. If you're looking at the history of that field of nitrogen fixation, the first idea that people had was to use the legume system, where you have the formation of what we call these root nodules, and to transfer that to cereals. Another idea that also has been around for quite a long time is to transfer that nitrogenase from bacteria to plants. Can we have plants that fix their own nitrogen? That would solve all the problems about transfer of nitrogen from bacteria to plants. And in theory, it seems very elegant. There are several colleagues working on that kind of project. It's really difficult trying to express the nitrogenase into plant mitochondria or plant chloroplast. It's very complicated and lots of issues to address. There are approaches that are, in fact, much more short-term, and I'm going to talk about today briefly, is to modify the bacteria. So why don't we take bacteria that fix nitrogen close to plant roots and make them fix more, and force them to release that nitrogen, that ammonium, to the plant. That's entirely based on engineering the bacteria. And then there is a third approach, and another approach I'm going to talk about today, which is natural diversity. Can we use, in particular, plant natural diversity to find crops or progenitors of crops or close relatives of our current crops that would associate better naturally with some of these nitrogen-fixing bacteria? So today I'm going to cover briefly three topics: the engineering root nodule symbiosis, the engineering diazotrophs, so two approaches heavily based on genetic engineering that we are working on in my lab, as well as the natural diversity aspect also that I'm working on. So first, briefly, engineering root nodules, the legume system, into non-legumes, into cereals in particular. I really wanted to show you that cover page from that bulletin from 1917, basically exactly talking about that goal. So clearly it's not a novel idea, but we think now that we have the tool and knowledge to be able to make significant progress, and I'm going to show you why briefly. So there has been a lot of work over the last decades on that legume system, on how these rhizobia, nitrogen-fixing rhizobia, interact with and associate with legumes, and what are the, in particular, plant mechanisms and also bacterial mechanisms involved. So what was found over the last decades of work is that rhizobia, these bacteria, produce signals that we call nod factors. And these signals are perceived by receptors on the plant plasma membrane. And these receptors interact with the co-receptor, and that leads to a whole signaling pathway leading to the activation of specific transcription factor. One of these really important transcription factor, we call that NIN, And it's involved both in what we call the colonization of the plants, but also in what we call organogenesis, forming the structure, the house, if you want, the nodule. What we found, and not only my lab and many other labs for the last decades, is that these mechanisms that rhizobia use to interact with plants to be recognized and to form that what we call endosymbiosis, where the bacteria are literally inside of plant cells. Many of these mechanisms are very similar to another association that we call mycorrhizal association, which is also an endosymbiosis, where the microbe is inside of plant cells. And what was found is that these fungi called arbuscular mycorrhizal fungi produce very similar signals recognized by very similar receptors, strikingly connecting to the exact same co-receptor and activating that exact same what we call common symbiosis pathway. In legumes, if you make a mutant in any gene of that common symbiosis pathway, the plant become unable to associate with rhizobia, unable to associate with mycorrhizal fungi. And so clearly that the top part of my figure here is very similar between the two symbioses. From an evolutionary standpoint, the arbuscular mycorrhizal association is very ancient. It appeared with land plants, and many plants, including cereals, have that whole pathway. The association with rhizobia is much more recent from evolutionary standpoint. And so it's very tempting to think that what rhizobia did is in fact to mimic the signals of these fungi, and that whole pathway allowing an endosymbiosis has been co-opted from an association with fungi for rhizobia. The bottom part of my figure with the organogenesis forming the root nodule is very similar to mechanisms we find in many plants to form lateral roots. Lateral roots and nodules are organs that form on the root. They are lateral organs on roots, and their mechanisms are actually very similar. We find nin-like proteins involved in forming lateral roots with similar cytokinin receptors and very similar genes involved. So very likely the nodule organogenesis part, how to form the house, if you want, was co-opted from the program to form lateral roots. So the idea on how to create a new symbiosis really comes from a combination of our genetic knowledge and evolutionary knowledge. We know that many plants, legumes, but also cereals, can form mycorrhizal associations. And so let's say all the cereals, maize, sorghum, wheat, rice, they all have that program to associate with these fungi. They also have a program to form lateral roots. And so the idea is sort of to recreate what evolution did in legumes and allow in a non-legume host, like a cereal, rhizobia to be recognized with these receptors, or to modify the signal produced by rhizobia to ultimately activate that common symbiosis pathway and allow endosymbiosis. And the idea is to recruit the lateral root program already existing in cereals to form nodules. So to show you that it's not completely crazy, I'm going to show you just one example. But before that, I want to mention that there are basically two groups working currently on that kind of project. You have the ENSA project led by my colleague, Giles Oldroyd in the UK and funded by the Gates Foundation. And then we have a project in the US funded by the Department of Energy between my lab and colleagues at the University of Florida. The approach is the same. The only difference is the model that we use. In the ENSA project, they use barley. They use a cereal right away. In that NITFIX project in the US, we use poplar as, I would say, stepping stone towards cereal. Poplar is closer to legumes. It's not a legume, clearly, never evolved nodulation, but it's much easier system to engineer. And so we take that as a first step. And to show you that it's not completely crazy, these are structures that we call currently in the lab pseudonodules that develop by modifying just a few genes and with the right promoter in poplar. And so they are very similar to legume nodules. They have a vasculature like nodules, and they are formed just by engineering a few key genes to trigger these structures. So at this point, I think we are very close to have the organogenesis nailed, to have the house, if you want, for these rhizobia. The challenge for us now is to engineer the endosymbiosis, and that's what we're working on. So that's one approach. Can we form a nodule on cereals? And as I said, probably doable, but it's we are not there yet, clearly. Another approach that I wanted to talk about, which is also based on genetic engineering, but which is much more short-term, is engineering bacteria. Bacterial genetics, as you know, bacterial synthetic biology is much faster than plants. And so that's why I think that approach is actually much faster. So for the bacterial side, the idea is, why don't we take nitrogen-fixing bacteria and modify these bacteria so that they fix more nitrogen and deliver nitrogen to the plant? For these projects, we take bacteria that naturally associate or found already in the rhizosphere of our crops. I work with bacteria, mostly isolated from maize and sorghum crops. And we can rely on decades of genetics in nitrogen-fixing bacteria from the community. So that's the beauty of it, that the Sanofi community has been working on the regulation of the nitrogenase activity and the expression of what we call NIF genes that control the synthesis of the nitrogenase for decades. And so the idea here is, can we deregulate the system to fix more nitrogen and give it to the plant? And the short answer is yes, and we already have pretty good success. So that's a project for me that was funded by the NSF initially, which is also a collaboration with a company called Pivot Bio. And so what we do is that we play on the regulation of the nitrogenase. Briefly, in Proteobacteria in particular and Gamma Proteobacteria in particular, you have two regulators of the expression of NIF genes, NIF-A, negative regulator, NIF-L, negative regulator, NIF-A, a positive regulator. So the idea is pretty simple. Why don't we remove NIF-L? We remove the negative regulator and we overexpress the positive regulator. That works. That leads to much higher expression of the nitrogenase. Another trick we use is to affect the glutamine synthase, to affect the ability of these bacteria to incorporate, to assimilate the nitrogen, the ammonium they just produced, and so that they can use it less than wild-type bacteria. So ammonium is going to build up in these bacteria and leak out of the cells. And we also modify the ability, we actually prevent these bacteria from recapturing the ammonium that leaks out of these cells. And with all these different types of modifications that we need, of course, to fine tune, we can lead to what we call these ammonium excreting bacteria. And Pivot Bio already commercializes, has a product on the market with some of these genetically remodeled nitrogen-fixing bacteria. So great approach, but how can we do better? What are the challenges of these approaches? The main challenge we face is the decrease of fitness. The more we engineer our strains to fix more nitrogen, that's my red line here, and deliver more nitrogen to the plant, the more their fitness is decreasing. Of course, it makes sense. These bacteria spend a lot of ATP energy fixing nitrogen. We prevent them from using that nitrogen. Their fitness is going to decrease a lot. A second issue that we face is the delivery of nitrogen to the plant. These bacteria live on the plant surface. Best case scenario, they are plant endophytes, but they are not, it's not an endosymbiosis like in legumes. So the exchange or the transfer of the nitrogen from the bacteria to the plant is limiting. Briefly, what are we working on? To solve the fitness issue or try to alleviate some of the fitness issue, we try to work with synthetic communities and better understand bacterial interaction in the rhizosphere. How do these weaker engineered strains interact with other bacteria in the rhizosphere? Can we find competitors? Can we find helpers in the rhizosphere? The transfer of ammonium, we got actually we have a paper which is going to be submitted very soon on showing that mycorrhizal fungi, in fact, help a lot the transfer of ammonium from these engineered bacteria to the plant. They can serve as sort of extension of the plant root system to capture ammonium from the engineered bacteria and to the plant. So again, we can get a little help from microbes in the rhizosphere for that. So that's the second approach. Both approaches, both engineering nodules for bacteria, are heavily based on transformation and genetic engineering. And as you know, in many parts of the world, these kind of technologies are not really acceptable. And so we are looking for can we find a faster way to be able to deliver nitrogen to crops, in particular in developing countries. And for that, the idea was to explore natural diversity and plant natural diversity. Can we find crops or close relative of our crops that associate better with nitrogen-fixing bacteria? So several years ago, I collaborated with colleagues at UC Davis, and in 2018, we reported the characterization of maize landraces. So it's really maize. It can be crossed with the maize we grow in the US. And these landraces obtain about half of their nitrogen from the air, which was pretty amazing. Here I show you some of the data published in 2018. We were using 15N natural abundance to evaluate how much which fraction of nitrogen in the plant comes from the air, comes from the atmosphere, so from biological from fixation. And we saw that during about 4 months, during the growing season, these plants were obtaining about half of their nitrogen from the air. We used many other techniques to confirm that. I refer to that paper and other papers we published after that. But briefly, the trick in that case here, the biological trick is that these plants produce lots of roots on the stem. These roots never touch the ground. We call them aerial roots. They stay in the air. And when it rains, these roots produce a lot of gel. And so that gel or mucilage is actually the place where nitrogen fixation takes place. And these are the data on the right, where we measure the nitrogenous activity in different parts of the plant. Even in these roots before rain, there is no fixation. And after rain, there is a lot of very high levels of fixation on these roots. And later we showed that really all the fixation happens in that gel produced by these roots. So it's a pretty amazing system that initially we characterized in maize from one village, but later, after not working with the Mars company anymore, I started working with the CIMMYT. And with the CIMMYT, which is an international center, we started surveying, looking for that phenotype in maize from around the Oaxaca, and we started finding it in many places. It's not only in maize from the Totontepec, it's also throughout the region. The trait is even in Teosinte, which is the progenitor of corn. I wanted to mention that the trait is also present in sorghum. We have 2 publications under review right now, but I wanted to mention that in 1986 already, colleagues at the ICRISAT in India had some hints that something was going on in these aerial roots and the gel produced by sorghum. I started working again in 2019 with these colleagues at the ICRISAT, and now we have that project funded by the Department of Energy that we call SONAR, where we actually showed literally a few months ago that these sorghum accession obtain 43% of nitrogen from the air, which is pretty large. All right, I wanted to finish and show you these kind of that summary slide with the different approaches. And a classical question I often get is, are we there yet? When are we going to have these technologies available? Well, the genetic engineering of the bacteria, it's already there. It can be, of course, improved, but you have the Pivot Bio company and probably other company using that genetic engineering of the bacteria, and it's already commercialized in the US. And the approach of breeding, using these crops that naturally associate well with nitrogen-fixing bacteria, the breeding has been well started, and I'm doing that in my lab for corn and sorghum. I'm saying 10, 20 years, but it's going to happen for sure. When are we going to get these nodules on corn? Really, I don't know. I'm putting 20 years, can be 30 years, can be more. The approach of engineering the nitrogenase into plants is even longer term and even more ambitious. So I'm almost hesitant to put a number of years on that one. But of course, it's exciting, but really long term. All right, I would like to finish and acknowledge funding. My lab is funded by NSF, USD, and the DOE. Also, some funding from corn growers and the Pivot Bio company. Thank you very much for your attention. I will be glad to take your questions.
Thank you so much, Jean-Michel. We have time for one question right now. Does anybody want to hop on? Ilva.
Unmute. There we go. Thank you so much. That was fascinating. I must admit, I've been quite skeptical to this whole line of research. I'm a soil ecologist, so you're always thinking like nature finds a way, and if it hasn't found it. But with CRISPR and all that, I can see an opening to these things. I was just curious, like those free living diazotropes, how much are they actually fixing, you know, compared to just what is ordinary there?
So how much nitrogen we can really deliver with the engineered one?
Yeah.
So the numbers are between 5 to 15% maximum. I would say 10% would be an average of nitrogen from the air delivered to corn using that kind of technology. Will we be able to go much higher? I'm not sure. There is, I think, some opportunity in combining that trait with the classical plant growth promoting traits. There is a synergy there, because if you have a larger root system and more of these bacteria delivering nitrogen in the root system, there is a synergy there. But at this point, I would say the numbers would say 7 to 10% of nitrogen from the air is, I think, is the fact. I mean, the data support that.
And just a quick follow-up with the poplars that you're working on, they're not currently fixing, but you know, I'm a mycorrhizal ecologist, so we you know, estimate that up to 20% of the carbon fixed can go to the fungi. So it's a substantial amount of carbon. Are you just kind of assuming then that the plants will upregulate photosynthesis and compensate for that, or what's the idea behind that?
That's a great question. That's for the poplar nodules. I don't know how it's going to happen. It's hard to guess. I'm a bit optimistic in that for the aerial root project, where we have the roots and the gel. We did some modeling and with a colleague at the University of Florida to calculate what's the carbon cost of a phenotype like that, and we estimated it's actually 2% of the carbon going into that kind of trait. And so I'm optimistic that 2% of carbon for a C4 crop like corn should actually have a sort of minimal impact on yield. It's all modeling at this point. We basically are still working on the introgressing the trait into corn that we grow in the Midwest, and so where we will really be able to quantify that properly. But I think the cost of the trait and the impact on yield is really going to depend a lot on the type of plant. And I'm optimistic that C4 plants like corn, sorghum, should be able to afford it. It may be more difficult for C3 plants like rice or poplar.
And what bacteria are they in the aerial roots? That's my last question, I promise, and then I'm done.
It's actually a wide range of many. It's quite diverse, but with a strong enrichment of protobacteria and a lot of gamma protobacteria like Enterobacter, Klebsiella, Pseudomonas.
Thank you.
They recruit almost whatever is in the environment. So the microbiome we get in Wisconsin in the gel is very different from what we get in Mexico. But in both cases, the plant can recruit nitrogen fixers from the environment.
Super interesting.
Thank you so much, Jean-Michel. We'll have time for more questions at the end, but now I'd like to introduce our final speaker, Dr. Lee DeHaan. Lee has been with the Land Institute for more than 20 years, exploring the potential for developing perennial grain crops from a wide array of candidate species. Since 2010, his attention has been dedicated to developing intermediate wheatgrass into a perennial grain crop using traditional breeding and genomic methods. Over the past 6 years, companies have begun to release food and beverage products from this wheatgrass under the trade name Kernza. And the program now has collaborators in the US, Canada, and Europe exploring an array of topics, including the ecosystem services of perennial grain crops, the functionality of Kernza in food products, disease management of Kernza, and optimal management of Kernza in diverse environments. Welcome, Lee.
Did the share work?
Yes.
All right, great.
We see it in presenter mode at the moment rather than...
Must have done the wrong share. Let's try again. How's that?
Yep, great.
All right, great, thank you. Yes, thank you so much for inviting me to talk today. I see a lot of synergies actually between the previous presentation and what I'm going to talk about today. It's exciting possibilities there. So the title I was given was breeding perennial crops, and I decided to restrict this a little bit to perennial grain crops where my expertise is, recognizing that perennial crops of all sorts have a lot of value for the topic at hand, but perennial grain crops have a kind of a unique twist on it. But I think a lot of what I'm going to say would be relevant to breeding perennial crops of any sort, including forages, including tree crops, etc. Just want to start with a little bit of background as to why we do this work. I want to mostly stay away from that and focus on what's happening around the world in perennial grain crop breeding. But just to quickly to say that the below ground structures present in perennial crops are different in their quantity and distribution, and especially temporal distribution relative to our annual grain crops. And we've come to understand that the presence of these deep extensive roots throughout the year are drivers of a lot of ecosystem changes and differences. There's just one slide showing the quantification of root density in intermediate wheatgrass, the crop that I work on, the perennial, compared to annual wheat grown in Michigan. Many times greater root presence, even at the height of the growth of the wheat in the year. And then for 5 months of the year, the wheat has virtually no roots living or present. So the presence of those roots do many things biologically. I wish I would have put up a few slides about the microbial changes that we've seen. So there's been work done comparing restoration of even perennial monocultures or bicultures, what happens to the microbial community relative to under an annual grain crop system. And we've seen that becomes a lot more like the soils under the native system. There's a lot of hope about restoring soil health and quality by having more roots in the soil. That applies to the cover crops, as was talked about earlier, but also perennial crops that can have extensive root systems present throughout the year, have extensive mycorrhizal systems. Nitrogen fixation has been mentioned. All these things happening more and happening at a greater scale with larger root systems present for most of the year. The interesting, strongest point that's come out of the research has really been efficiency of nitrogen use and nitrogen cycling. So globally, about 50% of nitrogen fertilizers obtained through the Haber-Bosch process are typically lost to the soil, through the soil to the water or into the air. With perennial restoration, even with high levels of fertilization, we typically see over 90% use, even up to 99% utilization or capture within the system rather than loss out of the system. So doing more with less. So I see a great synergy between the previous talk about enhancing biological nitrogen fixation and then also trying to capture that which you do have and not losing it out of the system. So with that background, why we're doing the work, there's kind of two major approaches that are happening in perennial grain crop development. I'm going to have a different presentation here, and then I'm going to talk about a lot of people's work around the world and not just my own work, but what's going on kind of globally in perennial grain development. And here's 6 crops that have pretty extensive work going on around the world. On the left-hand side are direct domestication projects where we're looking at taking a wild plant and turning it into one that can produce an edible grain for human consumption, versus those on the right where we're essentially modifying an existing annual crop, trying to make it become perennial and grow for many years. A little bit more about what those methods look like. On the left, again, the domestication process. On the right, we see a hybridization happening first between the wild perennial species and an annual crop. And there could be few different twists to this. Along the way of domestication, we can also utilize genes from annual crops. We can do that through gene editing or through wide hybridization, even knowledge of genes through gene editing. So, but with the wide hybridization approach, there's kind of 2 different attacks on that, where some species we kind of have to have an equal contribution from the wild perennial species and the annual crop. That's typical of the perennial wheat breeding that's been done so far, versus some other projects where maybe only a small amount of the genes present in the perennial species are being brought in the annual crop. I'm going to start digging into one of those a little bit more, and that's perennial rice, where the objective was to bring only what was necessary from the wild species. I'd like to point you toward looking, if you're interested in more information on this, the Nature Sustainability paper that came out this year, detailing the work that's been done on perennial rice. This is it started in the 1990s, was run at Erie, but really aggressively about a 15-year project in China with some support from the Land Institute and collaboration over the years. The development of perennial rice, starting with annual rice on the left, crossing with a perennial relative, Rhizolon gestaminata, very rhizomatous, long-lived species, obtaining kind of intermediate plants, and then generations of back crossing have led to these types of plants here shown upper right that can regrow after one harvest. You see at the base here last year's or the previous season's growth, regrowth, and then a second crop being produced. In the field, it looks like what you see across the bottom. First planting the bottom left, the sort of overwintering period where the plant has died back to the ground, and then a rapid regrowth and a second crop shown on the bottom right. And the exciting part about this has been how breeding has been able to increase the yield of these types over time. So starting at the bottom right of this graph from their publication, you see there's essentially 8 harvests of the red, the perennial line relative to the annual crop here. And so there's 2 harvests per year, a fall crop and a spring crop, the spring being lower for both the annual and perennial, oscillating back and forth, but about 8 harvests that were equivalent between the annual and perennial, and then the 9th and 10th. Starting to see that perennial genotype was falling off in its productivity. The grain produced is very similar to annual rice. It's sold into the market, indistinguishable from annual rice. And the farmers have been very excited about it in the region of China where this has been produced. You can see the number of sites and the number of hectares under cultivation has really increased as fast as they can increase the seed and get it out to farmers. Farmers are mostly interested due to extreme cost savings that they have from not having to repurchase and replant seed every year. But there was also demonstrated some early results showing benefits to soil carbon sequestration and enhanced soil quality as well from the perennial crop production. Moving on from rice to another more tropical crop, sorghum. There's a great potential to hybridize this wild perennial actually weed known as Johnson grass on the left, has a very strong rise in production with annual grain sorghum on the right. The hybrids have produced rhizomes that are much reduced in extent. So the perennial parent would have rhizomes extending out up to 6 or 10 feet within the first year. The hybrid plants have muchly reduced rhizomes, but you can see they grow very deep in the soil. That's how they can escape the cold. Even though the plant doesn't have much cold tolerance, it can go deep enough in the soil to avoid freezing and then regrow in the spring. It's a lot easier, therefore, to have a plant that overwinters in a more tropical environment. That work has begun now, moving some of these to Africa. Here's a slide showing a picture of Africa and location. The farmers and the researchers there are very excited about the potential, especially for drought tolerance, and ability to provide forage in the off season is really attractive. Been able to regrow in the dry season, whereas where annual grain sorghum has been produced, there's virtually nothing on that land. It's barren, so open to erosion and degradation, and also not providing any value of forage for livestock. So exciting opportunities. We think it's about a 10-year project, if well supported, to develop varieties that would be useful for farmers throughout certain regions of Africa. Next, I'm going to talk about a domestication project. This is the one I've led for about the last 20 years. Intermediate wheatgrass as a forage crop that's been grown, especially in the western part of the United States. So it's already useful for that purpose. And although when we first started this work, I said it was at least a 100-year project to get anything that would be really productive and useful for farmers, we've begun to see that the dual use nature, so farmers already growing the crop, already valuing the forage from it, can have an economically successful system if they can harvest forage and grain from it, and can use just conventional equipment as this farmer was doing in the field. So our limiting factor here is really grain yield, and we have to increase it to hit a target that's we hope to be similar in yield to wheat grown in the same regions. So for approximately the first 10 to 12 years, we worked through traditional breeding, which was growing the plants in the field, thousands of them in the field for multiple years, selecting maybe 100 of them, bringing them to the greenhouse, intermating them either randomly or targeted, then obtaining maybe up to 25,000 individual plants that would go back to the field and then be phenotyped again. So very labor intensive, but the big thing is it's very time expensive. So 3 years at least for this kind of system per breeding cycle. And you can see if that's your time frame, it's very hard to make progress rapidly. Through innovations in genetics, we've been able to move on for that and develop a genomic selection approach. So we still do this field evaluation for multiple years and multiple locations to get very good phenotypes, but those plants have all been genotyped previously, so we can use knowledge about the genetics of the plant and the phenotypes, combine them into a single genetic model, and apply this new genomic selection cycle here. So, the difference is that this cycle is the breeding cycle now, and it's only taking 6 months. We can do 2 generations in a year, so essentially 6 times faster than the phenotypic selection process. That shortcut just creates the opportunity now to begin to develop perennial crops in ways that previously would never have been possible, just because of the long time scales required for every breeding cycle. So we're growing about 5,000 seedlings, genotyping 4,000 of those, making selections out of those, intermediating them, and running this around and around as fast as we can. In the meantime, plants are going out to the field to continue that training that model. And exciting step we've taken over the last couple of years is now just to not have these space plants, but to put families of plants out in the field planted in dense plantings the way farmers would grow them. So we have a much more realistic environment to get the data to increase grain yield in a truly agronomic type system. Some of the traits we're looking at are the classic domestication traits, such as shattering resistance. Over and over again, we've seen that when crops are domesticated, one of the first and most important traits is that the plant holds on to its seed so that harvesters can come by and collect those seeds from the plant instead of having to sweep them up off the ground. Especially important today with mechanical harvesting. It's really surprised to find that we have found and really strongly shatter-resistant plants within 10 years of work. I took this picture from a strongly shattering plant, just tapping those heads on the table a couple of times, and all these seeds fell off. That's typical of a wild plant. Treated this one the same way, and no seeds were lost. You can also see the very large seeds, atypical of a wild perennial grass. So intermediate wheatgrass is one that grows along the roadside in many states, and you don't see seed of this size at all. So our breeding has dramatically increased seed size. Just want to show you sort of the trajectory through the genomic selection process over the last several years. So cycle 6 was the last phenotypic selection cycle, where we genotyped the plants and used them for training, and then moved on from there. And here you see a shatter rating where anything over 2 is becoming a serious problem in the field. Anything that's around 1.5 to 1 is pretty good at shatter resistance. And we're well on our way to our target. In fact, we're kind of relaxing selection on this trait because we're achieving a feasible level of shatter resistance at this point. You see we're kind of stabilizing here. Comparison, pre-threshing has been a trait that's kind of the opposite, is having the seed come out of the hulls. So the progenitors of wheat, if you're familiar with oats or barley, are also in hulls that have to be removed with processing if they're going to be used as edible seed. Intermediate wheatgrass was naturally hulled. When I started, less than 10% of the seed would come out of through threshing. And now we've achieved plants that are essentially 100% free threshing consistently. Again, it's a trait that we're meeting our target objective. We're able to relax that selection now. Finally, or another trait here, mass of seed per spike. With individual plants being evaluated instead of plots in this earlier breeding work, we were focusing on yield per head as a way to not just get more and more heads produced, but actually have greater fertility of the heads. So again, when I started the work, anything up to 0.2 was really high. And then as we began the genomic selection, 2.5 was about our average, and now we've more than doubled that. through genomic selection. So is there any ways to do shortcuts in this process? And we think there are breakthrough approaches that we can take. One of them is to use genes that are already accumulated in our domestic species. So here was an example of taking wheat, crossing it to intermediate wheatgrass, and now we're not really calling it a perennial wheat breeding program or hybrid breeding program, but instead more of the domestication, because we're back crossing to the perennial plant. So our objective is to only obtain a few genes from the annual in a primarily perennial background. And so this process of hybridization took embryo rescue, and the next generation, we did some back crossing to the perennial, and we'd only get one seed per plant, or even on thousands of florets might find one seed, and eventually recover fertility. And again, we have modern techniques that allow us to do what is not possible in the past. Perennial wheat breeding was begun in Russia in the 1920s and 30s, long before the tools were available to really achieve the objective. And now we have those tools available. So this was a skim sequencing approach, where we just look at the counts of number of reads on a chromosome. And up the top here, we have a piece of the chromosome set from intermediate wheatgrass. There's 21 of them here, down to number 21. These are all present. Then as we look down here at the durum wheat chromosomes, those all with no reads, those chromosomes are not present. And here's one chromosome, chromosome 3b, that is present from the wheat. So our objective has been to develop a panel of lines that have single chromosome additions from wheat into intermediate wheatgrass, so we can evaluate what the impact of the chromosome is, and eventually get down to maybe small linkage blocks intergressed from wheat into intermediate wheatgrass to bring in some important genes like seed size, shape, dwarfing, other traits that have been hard to get just in the wild species. Another advanced technique we're following is targeted mutations, using the knowledge that we have from wheat. Wheat has been sequenced, and there's a whole lot of information about gene function and domestication, not only wheat, but also other grains, rice, maize, barley, etc. The known genes that control important traits for domestication allowed us to develop a kind of a roadmap for editing a wild perennial grass into getting the traits that we might have desired that are already present in our domestic crops. So in this paper, we outlined a number of important domestication traits that were present in rice, wheat, barley, etc., and found their homologs in intermediate wheatgrass, and looked at them for potential targets. If we know the important region that changed in the domestication of the wild progenitor of wheat or barley, we can then target that same change in intermediate wheatgrass to obtain that same, hopefully same phenotypic change. This work is being led by Michael Palmgren in Denmark, and currently they have edited plants for many of these genes. The plants are being grown in the greenhouse. We don't yet have phenotypes in any of them, but the process of transformation of the intermediate wheatgrass has been successful. I was actually quite skeptical we could even do the CRISPR process on wild grass species like this, but bring in the experts from around the world, they were able to achieve that in just a couple of years, have a pretty reliable editing pipeline going. So we think it's quite feasible to achieve what once was kind of dreamed as maybe being impossible or unfeasible. The potential benefits to be able to have grain that's edible directly, so that our grain crops can come from fields that are as sustainable as our grasslands, and as those grasslands that gave rise to the deep, rich soils that we currently grow most of our grain crops on. If we can once again have crops there that have that same phenology of multiple year growth to build the soils again and build healthy production systems, we think there'll be great benefit, and we're really excited about what the future may hold. Welcome to take any questions.
Thank you, Lee. We have questions for Lee? Ilva?
Yeah, I guess the same question as I think Mike asked Virginia, like, are you looking at any below ground traits at all and soil health, variables?
Yeah, not me personally, but our team and our collaborator group is very large now. So definitely has been done and continues to be done in terms of especially the effect on soil and soil carbon. The exciting potential benefit for perennials is the amount of root biomass that's present and how that might translate into soil carbon storage and soil health. And that's unfortunately a really difficult trait to study because of the heterogeneity of soils and how important that is in the impact. So a lot of work going on in trying to model it and understand it more broadly and make it more broadly applicable. On the breeding side, where your question is the most interesting to me currently is what might be the impacts of breeding for grain yield on the below ground growth of these plants. So are we trading off large root production for more grain production? It's been known in wheat, for instance, that as we develop semi-dwarf wheat, when the tops got shorter, the bottoms, the roots also got shorter, and often because just the pleiotropic effects. So genes that control length above ground often control length below ground, and it might not be a trade-off in terms of carbon, but anytime you're changing above ground traits, you might expect to have some effect below ground. Overall, in intermediate wheatgrass domestication, the plants remain incredibly diverse. So the species is amazingly diverse. We've genotyped many plants at quite depth. So we have a good sense of the genomic diversity in our current breeding program, even though it's been through close to 20 years or 20 cycles of repeated selection. We still, when we quantify the allelic diversity, it still exceeds the entire germplasm of wheat. So it's 100 plants cycling every generation, and just because these wild perennials are incredibly diverse, we still have a lot of diversity remaining, and therefore below ground they look like everything still, and above ground they still look very, very diverse. So there's a lot to work with still. We haven't lost anything that we can't recover for sure, but it's going to be really interesting to do that. There's some breeders putting out mini rhizotron studies at every plant having genotypes and then having a mini rhizotron in that plant and trying to quantify what's going on below ground.
Yeah, thanks. Super interesting.
Rebecca?
Thank you. That was really fantastic, Lee. Thank you so much. I was at Erie back a million years ago when they started that perennial rice program. So it is really, really like thrilling to see that it's really getting out there into the world. And I was wondering why the initial years, the performance of the perennial exceeds that of the annual, apparently. But then it kind of over time,
yeah,
I can see that with the price. But any idea why it starts higher and goes lower over that time?
No, I don't think the researchers in China had any clear explanation for that, other than it's a different variety that GYE and things are, a lot of weird things happen. So I probably wouldn't draw too much from that.
Okay.
Just that any way that a certain variety is suited. So these weren't like near isogenic lines. They're different varieties. They're the most productive current rice variety in that region put up against the perennial rice variety, and every year things are going to play out a little bit differently. So basically, they're in the same ballpark as what I would.
Yeah, it's fantabulous. Any sense of whether or not the gene editing is going to be kind of accepted by the regulatory or other publics for tweaking the perennialization genes?
Yeah, it's a really interesting area to discuss, but and it's we're thinking about actually trying to get some some grant support to go to look into that more and bring in a broader team to try to answer that question from many different perspectives. The Denmark group is very interesting because they are working in a country and region where it's not allowable really for any purpose. And so they kind of have a kind of a circuitous route that they're taking, at least in their grant writing, which is to say that they're using the gene editing to prove that a certain gene will have the function that they want. So the gene editing is easier. And then after that, they're just going to use a mutagenesis approach where essentially you mutagenize a large number of plants, you sequence them and develop a library that you then search for the mutation that you want. And so there's a technique that they have of essentially developing this index system where it's lower cost than you might think to develop that index, but essentially you try to make every known mutation out there and then you go and get it by
clean it all up.
A legal means, but to obtain exactly the same product. So it's a method versus the outcome. So whereas it's quite possible that we could imagine the edited version could be brought to the US or other locations and planted directly and used. The whole, the patents and all of that issue is another huge one. But there is a group in the EU who's trying to look at the ethics of editing, and that's outside my realm altogether. But they're trying to lay out some scenarios where they could imagine clear ethical benefit or a clearly ethical case for where it would make sense to do gene editing. And they're really interested in this perennialization as one of their case studies, as a case where the benefit to humanity is clear enough in their mind that it could justify a technology that might be questionable for other reasons. So there's a lot to think about there, but it could make its way even into the EU as the approach that's talked about. And the idea is to really not force it, but to get the community to embrace it before you do it, I think.
We'll take a question from Sarah, and then we'll open up the conversation for all three of our speakers. So Sarah.
Okay, thanks. That was a great presentation. I wondered, you mentioned a lot of the different benefits coming from the various perennial crops that you sort of did the case studies of, not just soil health improvements, but management cost savings and dual crops and off-season production. How would you, could you characterize a little bit which of those benefits looms largest in different conversations, like when you're talking with growers or with funders or researchers, right, or just sort of what is the state of the conversation?
Yeah, right. Yeah, I'd say that when we talk to funders, what we hear the most is climate, and it's also when we talk to industry. So the buyers of grains are most interested in trying to reduce their carbon footprint, and they want to have a positive offset in some way. And so that's the story that's the strongest. The tricky part there is that consumers don't seem to be necessarily on the same page. So there's some products on the market that are marketed as climate beneficial, and trying to probe that and see whether consumers are willing to buy things that are benefiting climate or not. That seems to be more of a stretch. So it's more like the companies marketing themselves as being climate friendly more than they are marketing products that way. Their investors are demanding them to be climate neutral, for instance, more than that translating into consumers. When we talk to farmers, most of them are going to be growing. Currently, it's Kernza that they're interested in the U.S. That's the only one that's available for farmers to grow, and we're seeing those that are succeeding the best have the ability to use the dual use forage grazing. And so there's places where forage is really scarce during the winter at times of the year where this crop can really be helpful to them. And so they can have that benefit. And then secondly, when things go well and you can get a grain crop as well, that's a huge bonus. And so it makes the crop more attractive in that maybe you're almost certainly going to succeed on the forage side, and maybe you kind of really succeed and get a grain crop and make a few years where you have a really good income from the crop. So there's all those things. And there's also currently federal government money to support perennial grain crop planting. And so there's farmer interest around that, whereas conservation, it's eligible for conservation payments. So that's attracting farmers as well. And of course, I already mentioned the China, it's all about the economics of perennial rice. I should say organic producers in the US are really excited for weed control. So organic weed control, no-till has always been a dream that's seldom realized. And so the idea that you can have this perennial Kernza crop once established, establishment phase is tricky, but once you have it established, it excludes the weeds quite well, especially further west where water is a little more scarce. It pretty much excludes the weeds for many years without issues. So you can have an organic weed control system, even if it's in the organic rotation, something to break up your annual crop rotation that can escape those weed problems that you might have. That's where organic farmers are very excited.
Thank you.
We now have about 15 minutes for questions for all of our speakers. Do our speakers have questions for each other? Sounded like there were a lot of synergies between your work. Sarah.
Sorry, I can ask a question. It's maybe a little bit specific or esoteric or something, but it's mainly for Jean-Michel, but I'm interested maybe from all of our speakers about how you are viewing the area of work you're performing in the broader context of sustainable agricultural production and the committee's charge of the intersection of soil health and human health. And I think on the nitrogen fixation, the ultimate, the rationale is incredibly clear given our dependence on industrial fixation of nitrogen. But are there other sort of systemic production system or environmental type benefits that you look at in your breeding programs and research programs that you talk about, other than just the big, like, we can't chemically or use chemical fertilizer forever?
Yes. I also look at the benefits of arbuscular mycorrhizal fungi for carbon sequestration and drought tolerance. So these fungi are well known to help the plant for in case of drought, and it's doing that in two ways, both by helping to take up actively water from the soil with transporters, but also by modifying the soil structure. And there has been that long story about what is called glomalin as something produced by mycorrhizal fungi. Nobody really knew what that substance was. So that we're looking into what is that glue that's produced by mycorrhizal fungi for, and that affects soil structure, texture. It's different. It's a long-term benefit because when you think about modifying the soil, it's not necessarily short-term benefit for the grower. But it's also the benefit clearly of carbon sequestration. There is a beautiful paper from Toby Kearse published recently quantifying how much carbon is basically sequestered by mycorrhizal application, and the numbers are pretty impressive.
I can also try to answer that a little bit. Currently the largest driver of perennial grain research related to human health is really nitrate contamination of ground wells in the upper Midwest. So I don't have the map of Minnesota with all the contaminated wells, but it's shocking when you see it. And the push is coming from the state government and also from water wellhead protection zones throughout the state. So that these cities that have wellhead protection areas around their city well that are trying to keep it from having to be processed at huge cost. They're trying to keep their water clean enough. They're trying to only plant perennials around those wells, and so they're in some cases giving low cost or even free use of land to farmers who are willing to grow a perennial grain crop on that land to see if it can help mitigate the contamination of wells with nitrate. So that's in the clearest study we can look at various approaches to sampling water below annuals versus perennials, and it's just the most overwhelming thing about how much nitrogen nitrate leaches out the bottom of them. In terms of other nutrients, there's interest in again the mycorrhizal fungi and their uptake and acquisition of nutrients, and also the deep roots throughout the year. Are they taking up more nutrients? And we can look at some of the nutrient contents in these plants, but there are different species, and it's really hard to say what's being driven by the perenniality versus it's just a different species. But it's from the diversification point of the landscape, having a plant that is different. So perennials are good, but also just different plants are good. So there's kind of a double whammy there. If you can bring more diversity, you can diversify the crops that are feeding humans. And so the argument for Kernza among those who are trying to make products out of it is they're talking about the greater amount of fiber, the greater amount of protein, lower content of carbs, and the benefit of those things to human diet. So, and it just so happens that it seems that the fiber in intermediate wheatgrass doesn't taste the same way that wheat fiber does. So you can put more of it in the same product and it tastes better to a consumer. And so you can have a product that's perhaps more pleasing with greater health benefits as we're trying to move the human diet away from so many such carb concentration. So it's like a human health benefit. It's not specifically to perennial, but it's about diversification and getting something besides corn and soybeans and wheat into our diets.
Yeah, I would echo a lot of what Lee's said already. A major focus for us is crop diversification and having living cover on the landscape for as large percent of the year as possible. And I would say we haven't framed our work as much around human health. A lot of the crops that we're working on are cover crops or perennial forages that humans aren't directly consuming, but increasingly we are working on food crops as well, with dry beans and hemp. So there's a link there for sure. And then the well contamination issues that Lee is talking about are super relevant for sure with cover crops and our other perennial crops that we work on too. Yeah, thanks. Good question.
Any other questions for our speakers? Rebecca.
With that question of the higher efficiency of capturing nitrogen rather than letting it leach off and trash the environment, that seems like a big deal. Does it allow to use a lot less nitrogen on the one hand? I mean, I really love the idea of Jean-Michel's. I mean, the synergy among what everybody's talking about here is really striking, and I can see the better agriculture emerging from what you're saying, but I'm wondering about the paths. I mean, what are the incentives? Like, is that lower requirement of nitrogen going to translate into a strong motivation, and that lower seed cost, obviously, if you get to keep it same plants going?
Yeah, definitely. So like terms of production, we're harvesting a lot of biomass from those systems as well as grain. So the nitrogen export is pretty good, and the nitrogen fertilization rate has been around if it's being fertilized. So people are excited about intercropping with legumes as being one way to get nitrogen into these systems. And that whole question of if it's more efficient, can we get by with symbiotic nitrogen fixation as our primary source through an intercrop? There's obviously some cost to that as you're taking up space with the intercrop. Virginia has the good take on trying to breed them for being separate, but there remains some overlap for the most part. And so the idea of course, getting nitrogen fixation to happen more in the grain crop directly is exciting, but the system could work also with harvest of the perennial forage alfalfa, say, with a different harvesting mechanism. So there's currently a USDA grant funding the development of an inter-row harvester to drive through between the rows of your grain crop, perhaps, and cut off your alfalfa at the right time. Otherwise, when you're taking a grain crop like Kernza, you're waiting for 2 months longer than you should have taken your first cutting. And so the quality of the forage is reduced, the quantity of the forage is reduced. So having a system, and that is sort of an engineering solution, is exciting, because that can be a relatively quick thing. You don't have to do breeding for many, many years. If you can build a machine to make something happen, that's pretty cool. So finding some ways around that could be applicable to a lot of different ways of intercropping, even just annuals, but also annuals and perennials. So
I just want to say that our goal for the US is to be able to keep the same yield, maintain the same yield, and reduce the amount of fertilizer applied. Currently, on a very practical basis, you need about 150, 160 pounds of nitrogen per acre to grow corn. Our goal is to be able to reduce that by 40 pounds. So that the fact that we would need only 120, 110 pounds per acre instead of 150. So that's the goal. Reduce the application of synthetic fertilizer and maintain the yield. When it comes to developing countries, the target is a bit different, because there they have very limited input. So I would say it's almost easier, I think, for us to make progress for developing countries, because they start from very low.
Silva?
Yeah, Lee was getting at something, and it's a question that is only loosely formed in my mind, so it may not be well structured. But what do you think about the relative importance of breeding versus just adopting, crops and that we already have? And if so, like what are the what is hindering us doing it, and what could incentives be to increase adoption? Because part of this report is also to come up with recommendations. And how large is breeding in all of this? It's clear if you have a wheat that you can engineer to fix nitrogen, that could be a game changer, but in the grand scheme of things, I'm curious about your thoughts on that.
I guess from my perspective, like, there is a lot of good management research already that demonstrates a lot of these systems can work, and I think that breeding can help to enhance productivity, or I talking about like reduce some of the challenges that are keeping farmers from adopting the systems currently. But I don't think, like, breeding doesn't necessarily have a silver bullet for the solutions. And I think, I think there's contributions there, but there's also these sort of policy incentives and and developing markets for the different crops, and you need all of it, and breeding is.
Yeah, I think you were nicely highlighting some of those incentives and hindrances, and
I wanted to mention that in terms of challenges, I think one challenge for some of these phenotypes is just how difficult they are to measure, to quantify in high throughput in field conditions. And so again, coming back to my nitrogen fixation, it's one limitation is that the techniques are extremely expensive, and looking into ways to increase the throughput, find faster ways to do that in the field. But that's the problem. These are difficult phenotypes, just like Virginie and Lee were mentioning. For root phenotypes, they are just very difficult. So if you can find better ways to measure that in high throughput in field conditions, that would be a game changer.
Mm.
The way I approach sort of management versus breeding is, often breeding is an elegant solution, but it takes time to get it. So in the meantime, you got to do some other stuff, and maybe it's so expensive to do the breeding that you don't do it at all. But when it comes to disease resistance, you can have resistance bred in, or you can spray an herbicide or spray a fungicide, and the breeding is more elegant solution to the problem. But the other way to talk about it is analogy of sort of hardware and software, that software is like the management that we do in agriculture, and the plants we grow are kind of the hardware. So the system's often hardware limited. So there's some things you can't do with software. You can't have a miniature cell phone computer in your palm just through software on a PC, right? It doesn't turn into a little computer, but that's a hardware thing. And so we're kind of the same thing in agriculture today, that a lot of the investment, agricultural research investment, is on management solutions, because they tend to be a little quicker. They fit it nicely into our 3-year research timelines. You can write a nice proposal, you can get a nice result, you can have a package that you can deliver at the end of it. And breeding solutions they might take 10, 20 years. It might take a person's lifetime. And it's hard to get that funding behind that. And so you kind of have to have an emergency often. There's some disease that's going to, result in starvation. Then we'll throw a bunch of money into breeding, but we don't often use it to solve the solutions that that breeding can solve, but it hasn't been brought there because the time and the commitment over time is just too hard for society to get behind it. And what's exciting now, I think, is that we're starting to see our society coming around to thinking about food in a different way. We're more willing to think about our food production as having to deliver multiple benefits. We want our landscapes to be healthy and clean. I mean, the title of your project here is part of that shift in the way that Americans are kind of seeing their food and seeing agricultural production. So asking farmers to deliver things other than food also asks agricultural research organizations to deliver different kind of crops to the farmers for them to grow. So if we just put it on the farmer's back and say, grow it differently, and even if they're growing with the best methods and they're still polluting the wells in their region, and that's happening even with best management practices, wells are being polluted. And there's a paper that came out a year or two ago saying, with best management, we're still going to have undrinkable drinking water. There's no current solution. We need something new. And so that's where breeding is needed to provide the kind of thing where management is just coming up against what's impossible. So that's where it's really exciting. And then the thought that you can bring those two things together, it's not just the hardware, it's also the software. So new management systems and new genotypes, tremendous breakthroughs when we get to that level. So,
and recognizing those services and having the farmers be paid for those services.
Yes, exactly.
Right. Yeah. Thank you.
Virginia, Jean-Michel, and Lee, thank you so much for joining us today and helping the committee think about its statement of task. And thank you all who have joined us online. The committee does plan to hold one more webinar on October 23rd, and more information about that will be forthcoming on the committee's website, on the study's website. And until now, we are adjourned, and thank you again for attending today's session.